Okay guys, let's take a look at the genetic code. So the genetic code is what's going to be shown here, and the genetic code representing our code for our messenger RNA. The specific sequence, uracil, 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 that's what's going to code for this particular amino acid, which is phenylalanine. So there are 20 different possible amino acids, and so you can see all the different amino acids shown here. Is the difference between the start versus the stop codons. So the messenger RNA will have AUG for a start codon. A mnemonic I like to use is like school starts in August. So you have AUG, and then these are all of the different stop codons. So once we have this particular coding for that messenger RNA, we will release that new polypeptide strand that we've just made. So a mnemonic I like to use for this one is you are gone, you go away, and then you are away. So one of the things that we talked about was a start codon. And so if we have a messenger RNA that has AUG, what would be the codon, the anticodon that's going to be represented here by this tRNA? So tRNA is going to be what carries the amino acid with it in order for it to bind into our ribosome. And so it's just the complementary base pairing. So you'd have, instead of, remember we're substituting that T, so it'd be AU, UA, and then this here, for this tRNA, you would also have C here. And so this is go for our methionine amino acid. And so let's look at these different steps here. So step one is initiation. So initiation, we have our tRNA complex here. And then we also have an enzyme which is known as synthetase. So synthetase is going to be the enzyme that's transporting this tRNA, and we transport it here to the ribosome. So the ribosome has a small subunit, and then this is the large subunit. And this is what's representing our messenger RNA. And so there's 3 sites within this ribosome, the E, the P, and then the A site. So during initiation, we have that first tRNA that gets transported into the P site. The first thing that happens before the large subunit binds is you have to have the small subunit, the messenger RNA, as well as the tRNA. Once you have that complex, then that large subunit will then come over the top. This is that initiation stage there, but then we then move on to elongation. For translation, look at the steps. It's initiation, elongation, and termination, and these are the same steps required for transcription. But back to elongation, so the first step is codon recognition, so that is going to be step 2A. And then we now can get the movement of our tRNA coding for a particular amino acid, which will enter into the A site. So it accepts at the A site, and then we move on to step 2B, where we get the formation of our peptide bond. And then that tRNA has to exit. So it's going to exit through the E site. So that's what happens during step 2C, which is translocation. So that ribosome moves in that direction, and then we get the removal of that tRNA, and then that process will continue to happen where we get that tRNA with the amino acid here into the A site, and then you get that peptide bond formation, and then the movement of that tRNA out. So then the last step, so this was translocation, the last step is step 3, which is termination. What would this be? What code would that be? So it'd be one of our stop codons at that mRNA. So for instance, you are away. Binding into the A site, that is our release factor. That is our release factor in order to release our polypeptide or our protein, depending on how many amino acids are within.